And today I just like to talk about evolution, but obviously in the context of cancer and cancer evolution. I'll try and take you from this very sort of basic principle level to also what it might mean for patients, etc. So just to start with a disclaimer, I am not a population geneticist, but I do, of course, study cancer evolution. And when I was preparing this talk, I was thinking to myself, okay, I'll look up the program and I'll see, what I can learn about you guys and how I can overlap what I do with what you guys do. And I saw this excerpt on the website that had like a short description about what population genetics was. And I saw a couple of sentences in this excerpt and I thought to myself, wow, that's really relevant for cancer evolution. So one of them is that population genetics seeks to describe the distribution of and the change in allele frequencies in natural and domestic populations. And if you think about this in terms of cancer, is this true? It absolutely is, but it's in terms of cancer cell populations. So there's a definite overlap there. So we take that box. It attempts to explain the evolutionary phenomena such as adaptation, differentiation, and speciation. Well, do we try and understand adaptation? Absolutely. This is typically in the context of therapy. Do we try and understand speciation? I would say kind of yes. We have our own version of speciation, but I'll refer to that later on. I think differentiation maybe means something a bit different here, so we'll slightly skip over that, but we can tick this box for the most part. It's also interested in the interplay of genotypic and phenotypic level phenomena. This again, of course, is absolutely true. In cancer, they evolve because the phenotype changes. So we're also very much interested in how the genotype and the phenotype interact. And then we have this last sentence: a steadily growing number of fully sequenced genomes are available, and the importance of natural variation has been recognized by such genome projects. As you can imagine, this is 100% true in cancer research. Over the past 5 to 10 years, there's really been many studies that have sequenced thousands and thousands of cancer genomes. And from these, we've actually really inferred new insights in terms of how they evolve. So we can take that as well. So just to give you a sort of brief introduction of the whole talk in total, I'll give an introduction to cancer evolution. I'll do this a little bit longer than I normally do to really try and get us all on the same page. So I'll talk about some of the concepts, some of the terms, and some of the ways we actually analyze cancer evolution. And then I'll show you some examples from lots of different research projects. I'll just show you some short stories, essentially, about how we can use these principles to understand cancer. And also be mostly focused on my postdoc work. So big credit goes to my two postdoc supervisors, Andreas Othariva and Trevor Graham, with whom I worked with during my time at the Institute of Cancer Research. And then I'll give some more perspectives and conclusions about maybe contrasting population genetics with cancer evolution. So I also included this slide just as a clarifying point of when we think of evolution and when we think of cancer. I think you can sort of split this into two different study groups, so to speak. One of them is the evolution of cancer. I suppose this is interested in, when did cancer arise? When did it become a problem for multicellular organisms? Why are different species more susceptible to cancer and others not? And you might have heard of something like Peto's paradox, which is this idea of, well, why is it a blue whale is not riddled with so much more cancer than a human, considering they have so many cells in their body, and you might naively assume that every cell has an equal chance of forming cancer. This is not my expertise, and I will not be talking about this today, unfortunately, so sorry to disappoint you if that's a problem. I'll be talking about somatic evolution. So this is referring then to the evolution that's occurring after the point of fertilization in a single individual, so within that particular individual. And this, of course, underpins cancer development. It also underpins the aging process as well. So normal cells mutate as well as cancer cells. And it, of course, allows cancers to adapt and change over time and change to different selective pressures. So here is a very classic diagram that we often show when we try and describe what we mean by somatic evolution in cancer. It might look quite similar to how you might imagine evolution in bacteria, for example. But the cancer explanation for this process is that, within our bodies, we have many cells, and even normal cells, they probably mutate a little bit every time that they divide. And of course, this is changed by, if a person chooses to smoke, if a person is exposed to lots of UV light, etc. But even still, if you, remove all these carcinogens, there's also an inherent process where, somatic cells tend to acquire mutations. And most of these mutations don't actually really do a great deal to the phenotypes of these cells, but occasionally you'll get an important mutation that hits a gene that is somewhat oncogenic. And then the cells continue to divide and acquire additional mutations, and then maybe you get a second hit. And it's through this acquisition of oncogenic mutations that eventually you form a tumor. But of course, this process doesn't stop at this point. the tumor is formed and it continues to divide, and it might actually acquire additional oncogenic mutations within this tumor population. And then it gets to the point where you observe this tumor. So typically a patient might have a tumor that's large enough to be detected clinically, and then you might have typically surgery. And this is the point at which we typically observe these tumors. And we are appreciating more and more now that they are really a mixed population that's been formed by this branching evolution process. And what we often do at this point is we then try and trace backwards how this process has occurred by looking at all the different mutations we see within these tumors. But of course, this is not ending at this point. Unfortunately, surgery is not often 100% effective. There's probably still some residual cells, and they'll go on to continue to divide, to continue to evolve, and to also adapt to different therapies, and maybe even also go on to form things like metastases, where they colonize essentially different parts of the body. But clearly, somatic or cancer is formed by an evolutionary process. So what does this mean for the nature of cancer? I think this is really important, and something we're appreciating more and more is that it really evolves through this stochastic process of mutational accumulation, and maybe something like a deterministic process selection, where if you have a certain gene that's selected in a certain cell type in a certain context, maybe it always has a certain fitness effect, although our evidence for this is actually probably somewhat lacking. But this stochastic process of mutational accumulation means that truly every cancer is unique. So both across different cancer types, but also certainly within cancer types, no two cancers are the same. They typically have completely different sets of mutations. They might share some key driver mutations, but also these can be very diverse. It also means that cancer is very adaptable. And of course, this makes it hard to treat because you have millions of cells, and these cells might have something that can fight the treatment that you choose to use. So be it, it's a very similar analogy to antibiotic resistance in a bacterial population, that you can have pre-existing bacteria that are resistant to that antibiotic. It's completely the same principle in cancer with therapy. And it also allows cells to adapt, like I've alluded to already, to completely new environments. For example, metastasis, where suddenly a cell might meet completely new selective pressures, new amount of oxygen in an environment, new mechanistic stresses, as well as it colonizes maybe to something like the liver or the lung, or a completely different site to where it began. So just to, again, sort of describe in a bit more detail this concept of driver mutation. So generally, I think we think that driver mutations are positively selected, and these come in the form of oncogenes, where a mutation typically leads to its overactivation, or these driver mutations can also affect tumor suppressors, where typically these will function to repress certain oncogenic traits, and these are knocked out. But we typically believe these to be positively selected, and they're accumulated over the process of the life history of a tumor. Far outstripping the driver mutations are what we refer to as passenger mutations. I mean, the ratio of driver to passenger mutations could be 1,000 times more passenger mutations than you have driver mutations. And typically, we think that these are neutral, although this is an ongoing area of research. Cancers often arise from fairly specialized cells that are already quite differentiated. So they don't have the responsibility to go on and form every cell type within a human body. So actually, potentially, there's a lot of genes that are not really that important to a cancer cell anymore. Unlike, for example, in the evolution of species or whole individuals and whole organisms. And there's one quite nice study from the Sanger Institute that showed this with the NDS analysis, which I'm sure you're all familiar with, the ratio of non-synonymous to synonymous SNPs as a sign for selection. And what they quite nicely showed is that in species evolution, when they looked at genome-wide at the different SNPs, they saw that the NDS was very low, indicating strong negative selection, which to me naively makes sense because these genomes are already quite well refined, and changing these these genes that have evolved over millions of years, you probably have to find a really unique mutation to give a selective advantage. But in cancer, when we do DNDS analysis to all these mutations that we observe, we tend to get either neutral DNDS or slightly or relatively highly positive selected DNDS for different types of mutations like missense and nonsense mutations. Just a brief, I know this is all relatively simple, and also from talking to you guys, I already appreciate sort of the background a little bit more now, but I thought I would also touch on the typical types of mutations that we have in cancer. So of course, we have single nucleotide variants and small insertions and deletions. These are very well studied and focused on from whole genome sequencing studies. we very typically get large chromosomal alterations. So chromosomal instability is something that a lot of cancers develop, and this means that they're able to actually duplicate or delete whole chromosomes or arms or even just small parts of chromosomes. And this is also oncogenic and is a strong driving force in a lot of cancers too. So they generate whole new karyotypes quite often. Gene fusions are also important, where genes can come together and they have entirely new functions. And also one very interesting phenomenon as well for me too is whole genome duplication. So this happens in around about 40 to 50% of cancers. And essentially what we think is happening is that the cell is trying to divide and it duplicates its genome, but then it fails to actually divide and suddenly it has twice as much DNA content. And like I said, this is actually very common in cancer, and we're only really beginning to understand why it's so important. So how do we measure how cancers evolve? Well, I referred to it on the first slide that we're interested in allele frequencies. So for us, it's typically variant allele frequencies in a population of cells. And one of the typical assays that we, of course, use is whole genome sequencing. And I would say that cancer evolution really kicked off with the advent of next generation sequencing. There was a lot of papers in 2011, 2012 that really then applied whole genome sequencing to studying cancer evolution. One of them is shown on the right here. This was another publication from the Sanger Institute in 2012 where they had really deep whole genome sequencing. I mean, this is deep sequencing even for today, and this was 13 years ago now. And what they're trying to show here is the different allele frequencies of the looking at the somatic mutations and their allele frequencies in the population and trying to figure out what subpopulations are present and how they're evolving. But it also can be measured by other methods. For example, you can trace lineage through DNA methylation changes. It's something that's sometimes done, and also using SNP arrays. So you don't necessarily have to use, sorry, I'm afraid that the text is a little bit moved, unfortunately, from switching laptops. But SNP arrays are essentially a non-sequencing based method where you have an array that has oligonucleotides, and you actually just try and find a signal of DNA content from different parts of the genome. So you don't necessarily have to use sequencing. So typically in terms of our experimental design for cancer evolution, I think there's primarily three different methods. One of them is to do a bulk analysis. So this is you typically do this because it's in some ways the easiest option in terms of getting samples from the clinic. Is you get a big chunk of tumor, and then you apply your deep sequencing method, and within this big chunk of tumor, there is different subpopulations, and you look, like in the previous example, for different clusters of allele frequencies, and you try and find which different subpopulations are present. But I have to say this also requires some quite bespoke bioinformatic analysis, and it is actually really quite a tricky exercise, but it's certainly done by many groups and done really well. Another example that I actually use more typically is what we call multi-region sampling. So where we take advantage of the fact that a tumor has spatial constraints as it grows, and so different subpopulations are actually separated from one another spatially too. So we take different samples across this population and we can compare them to one another. And finally, more and more recently, we start to do single-cell DNA sequencing. So this is really the ultimate resolution where, of course, the cell is the unit of selection in cancer evolution, and this really gives us a fundamental insight, and it means that we don't necessarily have to do this deconvolution process. And of course, you can sort of mix these approaches as well. So one thing you could do, for example, is take multiple bulk samples, but also do a within bulk analysis as well. Of course, you can also measure evolution by looking over time. So measuring different clinical time points. So for example, at diagnosis and at relapse. And also if a patient has metastasis, then you can also compare different metastatic sites. So I also referred to it before as well about I kind of feel that we have kind of a version of a species in cancer evolution, or as close as we can refer to it. We call them subclones. I've tried to not use this term. I don't know if I have already, but I tried not to use this term until now. But this is basically the idea that there are certain subpopulations within this bulk that are different to the rest, and they somehow have a shared characteristic. Relatively poorly defined, but I think we all agree that these subpopulations that we call subclones must share the same lineage, so they must be genetically related to one another. Typically, they also must share the same driver mutations. So typically, a subclone might be defined that it has a new driver mutation that other cells in the tumor population do not have. And then we sort of theorize that they must therefore have somewhat of a similar fitness, or they must behave somewhat similarly. But this sort of process of defining these subpopulations is a little bit muddied by the fact that instability of mutation rate is really high in these cells. So every cell pretty much is unique, especially in a very advanced cancer. Uh, and also a point of terminology, but I realize you guys probably also use this terminology as well. But typically, when we talk about mutations that are present in every single cell, we call them clonal. And these are cells which are occurring before the most recent common ancestor in these populations. And I unfortunately have to skip the GIF, but what the GIF showed, if you saw a little snapshot, it was actually a computational simulation of the acquisition of a subclone. And essentially what we saw there was a variant allele frequency spectrum. And at some point, one of the cells acquired a driver mutation and suddenly had a selective advantage. And it was just a simulation of this new subclone basically sweeping the population because it begins to outcompete the other cells. And this is a process that we believe is, commonly occurring in the process of cancer evolution. So we also use phylogenetics in cancer evolution. So these are some overlaps that we're trying to find. There's certainly some very sophisticated methods out there, but I think, at least in my research, and I think it is also quite popular to use some maybe more simpler models, such as parsimony. So here on the left, I'm showing another very early cancer evolution study. This is one of the first to really use multi-region sequencing. And what they did is they did whole exome sequencing, I think it was, of all these different regions, and then asked what mutations were in these different regions, whether they were present, yes or no, and basically used this to generate a phylogenetic tree. And then we have some other algorithms that are maybe a little bit more bespoke to cancer. So as I mentioned already, chromosomal instability is really important in cancer evolution. And every sample that you take can potentially have a different copy number profile. And this can also tell you something about how these tumors are evolving. And we have different tools. For example, this one's called MEDIC2 that try and explain how these alterations are acquiring over time. And this tells you a little bit more as well about the relatedness of these different samples. And why is phylogenetics important? Well, once you've mapped this genetic lineage, you can also sometimes get a sort of tumor geography. So you can map back these different clades to where they actually were sampled from within a tumor. You can also, of course, get a sense of the order that mutations occurred in. And this is really important for understanding how cancers develop, because if you often see mutations that are occurring before the most recent common ancestor and are very early, you can be sure that they're very important in the early stages of cancer development. And of course, the opposite is true as well. If you see mutations that are subclonal and are not present in every cell, often maybe these are mutations that are being positively selected under treatment or are just generally important later on in the evolution of cancer. You can also use phylogenetics to trace how subclones behave and migrate in terms of metastasis and also over time. And you can also, from these phylogenetic trees, calculate some tree statistics that you might use to measure diversity and also evolution. And I'd just like to also briefly take a mention to stress the importance of tumor heterogeneity. So as I've alluded to already, there's a large inter-tumor heterogeneity, so every cancer is quite different from one another. But what we're obviously particularly interested in is intra-tumor heterogeneity, so within these tumors. And what does this mean from also a clinical perspective? So it means that genetic biomarkers that might be predictive of how patients are doing are not necessarily in every cell within that tumor. And so you have a certain chance of sampling that cell or not. And this actually has impact in terms of therapy or of course, making predictions for how patients might behave in the future. And of course, one of the hypotheses that we have is that some of these tumors that are more diverse might also, of course, be more adaptable, and they might also have subclones that are already present that are resistant to therapy in the future. So that was my sort of whistle-stop tour of an introduction into cancer evolution. So now I'll show you some research, some kind of short stories across the past of different things I've been involved with. Like I said, mostly coming from my postdoc work. And a slight warning, you actually saw it already because I think we moved to that slide, but the next tumor picture is a little bit gory. So sorry about that. But in this study that we performed in colorectal cancer, for example, we wanted to apply this method of multi-region sampling and sequencing. And here in this very gory picture, you can see that we have different areas that we sampled from A to D. And actually within these different areas, we even then subsampled even more. And then on top of that, we also took individual glands where they existed in these samples. So in the normal colon, you get glands, and sometimes these still exist within the cancer tissue. And these are quite nice because they're kind of like clonal factories because all the cells are very closely related to one another. And in these different samples, we actually did some multi-omic sequencing. So we did on the not for every single sample, but for a large subset, DNA sequencing, RNA sequencing, and some chromatin assays as well, which I won't actually touch on. So what could we do with this information across this cohort of 30 patients? Well, one of the things that, of course, we can do is create phylogenetic trees and ask ourselves how these cancers are evolving. And you can see a nice example here of a lot of different samples from the tumor showing us the phylogenetic history of these tumors. And this is some work from my former colleague Timon Heider, who, through simulation work, wanted to understand, what these trees, they tell us a nice story, but what can we actually quantify from these trees? And so what he did is he created a computational simulation of how he thought the cancer was growing, and actually used this simulation to recreate the phylogenetic trees. And after many, many simulations, he essentially found parameters that fit the data very, very nicely. I think this is a particularly striking example of a good fit between the simulated tree on the right and the real tree on the left. And of course, once you've simulated the tree and you know how you simulated it, you can ask, okay, where is selection happening in this phylogenetic tree? And interestingly, in this case, we see an inferred selection event on the top, which is linked to a KRAS mutation. It makes complete sense. KRAS is a super oncogene and it's subclonal and it's clearly positively selected in this particular cancer. But interestingly, we also inferred a second selection event where we had no obvious oncogenic candidate for what was actually causing this selection event. So through these simulations, we were actually able to maybe try and look for more insights into what is driving selection in cancer. I mentioned as well that we also had matching RNA sequencing in this particular study, and this is work by another former colleague of mine, Jacob Hausen, who took this data. So we had our phylogenetic trees, and we knew the genetic lineage between these different samples. And we want to ask her, what does this mean when we compare it to the expression data, which we kind of used as a readout of cellular phenotype. And so we took a method actually inspired from evolutionary biology, phylogenetic signal, and used this to quantify, just how much these phylogenetic trees could explain the expression data that we had. And actually, we found very little evidence for heritable expression of genes. We actually saw that a lot of genes were not really explained by the phylogenetic structure, which actually kind of led us down the road of investigating plasticity in cancer cells and how actually their expression patterns can change regardless of their genetic background. Moving on to a separate study, this was in breast cancer. And in this study, we actually had two patients that had essentially donated their body to science. And at the point of death, when they died of their breast cancer, they underwent a rapid autopsy. And at this point, they had many metastases across the body. And in this rapid autopsy, what we were able to do is take samples from many of these different metastatic sites, and sometimes multiple different samples from these different metastatic sites. And then we applied deep whole genome sequencing across these different regions and did our classic evolutionary genomics analysis, so including copy number analysis and also analysis of different mutational clusters. And what we could do is reconstruct the phylogenetic history of these different metastatic sites. And there was a few things that really stuck out to us in this particular patient. So for example, here there's a clade that is within the liver that was really striking to us because it had an estrogen receptor mutation. And this patient was on essentially an estrogen blocker because their cancer was diagnosed as being dependent on estrogen to grow. So it made complete sense as a therapy to block this. But this mutation, essentially what it does is it makes estrogen signaling active regardless of estrogen being present or not. So it essentially renders the drug ineffective. And this was acquired essentially in the liver. What we also did is we looked at a blood sample that we also took from the patient at the point of death. And one thing that's quite useful actually in the blood is that you can detect tumor DNA. So when tumor cells die, they actually shed DNA into the blood, and you can take a reading of this. And this indicates cells that are releasing DNA into the blood. And we saw that many of the reads essentially that we were seeing in the blood match very closely to the liver. And so what we hypothesized is that in this patient, there was a tumor that had begun growing, or a subclone that had begun growing a lot, proliferating, and also dying, so turning over a lot, meaning it was detectable in the blood. And actually, the patient did die of liver failure, so it made sense as a real history of how this metastatic disease really occurred right until the very end stage. And we could also, using this phylogenetic tree, reconstruct a metastatic cascade and try and work out what we thought was the direction of travel of different subclones across the body. And also a nice thing that I sort of wanted to show to you in as a nice example of a kind of really extreme events that we sometimes see in cancer evolution. So across these different samples, we actually saw that they mostly had a kind of whole genome loss of heterozygosity. So most of the chromosomes had become homozygous. This at first was like quite surprising, and we thought there was something wrong with the data. But essentially, when we double checked the data and dived deep into it, what we were able to actually trace is that we think that they shared a most recent common ancestor, which underwent a haplodization event, then actually underwent a few cell divisions, and then underwent two parallel whole genome duplications and becoming diploid again. And then these two different lineages formed either the cells that we found in the primary or the lymph nodes or the other metastases. And this was super interesting to us. And how do we know this is the case? Well, in these few cell divisions between haploid and being diploid again, mutations were acquired, and we saw that they were doubled separately and completely separately from one another in these two different lineages. And obviously, after that point, everything was just in one copy again. That was our evidence for this very striking event. And interestingly, this seemed to immediately precede the metastatic event. So this was really important for what went on to then seed all these different tumors across the body. So another example as well of analyzing evolution, this time over time, again in the context of metastasis, going back to colorectal cancer. So we also had the study here where we had about 20 patients, and we sampled tumors from the colon and also from the liver, which is a very common point of metastasis, and as well as a couple of other organs too. And here we had very long-term data sometimes, so sometimes up to 6 years later, many different samples across this period, including really good clinical annotation about therapy as well and what treatments they received. And we could reconstruct our phylogenetic trees. And one thing that stuck out to us in a subset of patients, and what I'm showing here is essentially what's called the copy number profile. This indicates, which chromosomes are doubled and which chromosomes are lost based on essentially the total number of reads in that area. And we saw that in many cases, some of these profiles looked very similar over time. So, even if there was therapy in the meantime, even if the site in which you took the sample from changed from the colon to the liver or even to the lung, and even if you had therapy in the meantime. And this got us scratching our heads a little bit and asking ourselves, how could we explain this result? And one thing that we proposed in a recently updated bioRxiv paper is maybe this is an example in cancer of stabilizing selection, where you have a certain karyotype, which is really the optimum, and these extreme events of diverging away from the karyotype are kind of selected against. But this is, I think, one of the first times this has kind of been suggested, and we're still trying to really work out how strong our evidence is for this, but I think it is a really nice explanation. So we've presented there a couple of examples of observing in patients how evolution is happening, but how can we test, these ideas that we have of what we see in patients? And of course, right this very ironically, of course, you guys have heard of experimental evolution, and you're complete experts in experimental evolution compared to me. But I want to present you here just a short example of how we can actually perform experimental evolution in the context of cancer. So what we did in this particular study is we actually took a cancer cell line and we grew it in a dish, and we barcoded these cells, which essentially means that we kind of take a small random DNA sequence and insert it into individual single cells. And then we grew them out, and then we split them up into separate flasks. And for some of these flasks, we treated them with one drug, for some of them another drug, and for some of them were just our control samples. And what we wanted to do was actually treat them with a really high concentration of drug, so essentially a concentration that kills or basically introduces a 90% decrease in growth, to introduce a really strong bottleneck. And essentially what we did is we just left them and waited until they grew out and become resistant to what they were being treated with. And one nice thing that was done in this paper is we actually realized that in the media, there was potentially DNA that we could sample. So these cells are adherent and they stick to the bottom of the flask, but when they die, they detach. And they also spit out DNA into this media. So when you change the media, you actually have DNA that you can sample non-invasively from your experimental model. So this was a really nice way of actually tracking this over time in a non-invasive manner. And what we saw actually was with these barcodes and across these different replicates is that the same clone always was basically emerging in each replicate for each individual drug. And and it was also very, very reproducible. So we kind of saw the same growth patterns almost every time. also, it was clear to us from looking at the genetic data that this seemed to be driven by the same sort of genetic alterations. So in gefitinib, they had a MET amplification. In trametinib, they had a CDKN2A deletion. The sort of specific details don't matter, but for us, it was really clear that in some way evolution can be predictable within these experimental systems. Okay, so we've observed evolution in patients. We've also tried to test it, but does it have any value for patients? Is studying cancer evolution important for predicting how patients do? Well, this is something that we wanted to address in the study that we actually released last summer, where we wanted to study evolution in the context of a clinical trial. So this was, again, work from the Institute of Cancer Research, where we worked alongside the Royal Marsden Hospital, a cancer hospital that was associated with us. And there they performed a clinical trial in prostate cancer. And actually in prostate cancer at diagnosis, what they use is something called a 12-needle biopsy, which essentially goes into the prostate to try and find where the cancer is. And then they take out these different diagnostic needle biopsies, and they, in pathology, search for the cancer. But then what they did is they just then archived these as FFP blocks. And what we did is we went back and actually took this natural multi-region sampling and applied sequencing, as well as also taking images and applying an artificial intelligence method to do automatic grading, which I won't talk too much about today. So then we could, within a cohort of around about 110 patients plus, we had this genetic information taken from multiple regions. And we also had, of course, being a clinical trial, really good outcome data and follow-up data that was almost, I think, about 12 years on average follow-up. So then when we created this cohort of multi-region sequencing within these patients, what we want to ask is, from an evolutionary genomics or cancer evolutionary genomics perspective, what can we measure? Well, we did DNDS analysis and saw some relatively weak but significant evidence for positive selection of subclonal truncated mutations. We did some multi-region phylogenetic analysis using copy number alterations. So we did shallow whole genome sequencing because basically the quality of the data was quite poor and it was too poor to do deep whole genome sequencing, but you can get really nice copy number alteration data. And then we actually blinded ourselves to the outcome. We tried to do this in a sort of proper way, and we sat down and thought to ourselves, what different metrics of evolution can we measure before we unblind ourselves to how the patients did to try and avoid any bias? And we tried to think of a few, for example, how many events we were measuring in the phylogenetic tree, how many of these events were subclonal, what the percentage of the subclonality was, etc. We also had this measure which we called Spearman, which is essentially the Spearman's row of the copy number signal. We actually took 1 minus the Spearman row, so it became a measure of heterogeneity and actually poor correlation. And then we asked, okay, what does this mean in terms of patient outcome? And on the top, you can see two univariate analysis Kaplan-Meier curves for the number of events that we saw in the phylogenetic tree and also the copy number diversity, essentially. And what we saw is this was really nicely predictive of patients recurring quite a few years earlier in these prostate cancer samples from the point of diagnosis. And the really cool thing was, is that because it was a clinical trial, we also had all this important clinical information because, we have to also ask ourselves, does it have additional prognostic value on top of what's already used? So things like PSA, which is essentially an antigen in the blood that's used to measure the burden of different tumors, the grade group, so how dedifferentiated or how advanced the tissue looks, and also T-stage and N-stage are different forms of measuring just how big and how spread the tumor are. And when we plugged in some of the significant variants, covariates that we'd measured, these different metrics that we'd measured, we saw this copy number diversity also gave us some additional prognostic value. So it's clearly also important for predicting how patients did. And we sort of also tried to confirm this by, in some patients that recurred many years later, so in this case, it was about a decade later, sometimes we had even longer later. What we saw was that the clones that were emerging after all this therapy were very unstable and clearly have been formed by a process of a lot of instability, and it required a lot of more events. So this diversity that seemed to be driven by chromosomal instability might be really driving recurrence by allowing the formation of these highly altered genomes. But of course, you might be thinking, okay, this is prostate cancer, it's colorectal cancer, it's breast cancer. These are all really common adult cancers, but I work at the Children's Cancer Research Institute. So what about pediatric cancers? How are these any different? Do some of these insights hold up? While I will certainly argue that the evolution of pediatric cancers is quite different, and there's a few points here that try to summarize this, so clearly they evolve over a shorter period of time. So in prostate cancer, these patients are usually very old. But of course, in children, these cancers are developing over potentially months, but certainly not decades. For this reason, partially, they have fewer mutations because less time has elapsed in this period. They also don't tend to smoke, so they don't have such exposure to so many carcinogens. Interestingly, many of them are thought to arise during fetal development, so this is actually an incredibly new context for the evolution of a cancer, certainly in the early stages. And because of this, they probably have they do have a very unique landscape of driver mutations. But of course, we can still do a similar type of study in these tumors. So this is an example of a pediatric kidney cancer called Wilms tumor, which my lab is very interested in and I have been also working on in the past. This is an example where we applied multi-region sampling. This is not sequencing, this is SNP arrays that I referred to before, but it measures, again, the copy number profiles of these different chromosomes, where some have a higher copy number and some a lower copy number. And we can bring these different samples together and reconstruct a phylogenetic tree and really show that we can also see strong evidence of branching evolution in Wilms tumors too. And here is just some, again, little stories from different cases which we've studied in Wilms tumor. So sometimes in Wilms tumor we see some incredibly strong genetic diversity or genetic divergence. So in this particular case on the left, this tumor was sampled in 2 different areas. And when actually you looked at the histology, they had very different histology. It doesn't really matter what that histology is, but it was so different that they were kind of different subtypes. And when we looked at the genetic information, so this was SNP arrays plus targeted sequencing, we saw they also had mutations that really reflected what they looked like under the microscope in terms of their subtypes. So they almost looked like completely independent tumors. But when we dug a little bit deeper and did the phylogenetic analysis, what we actually saw was that they still all shared one single driver mutation in the beginning. So it seems like there was a driver mutation that created a pool of cells, and almost 2 independent tumors arose out of this initial pool of cells. And then also something that we quite strikingly see in Wilms tumor too is parallel evolution. So I believe this is very common in species evolution as well. But it's not that often seen in cancer evolution. So when we do see it, it's really, really quite striking. But in a particular subtype of Wilms tumor, there's a really strong late evolutionary selection for beta-catenin mutations. So much so that when we take several samples, we can often see many different versions of this mutation co-occurring in the same tumor. So this really tells us about the order and also the strength of selective pressure for these particular alterations. And of course, we also are interested in what this means for patients and whether we can use some of these measures to say something about how patients might do. So I took a very similar approach here as well by calculating these phylogenetic trees and asking how many different events we see in these phylogenetic trees as kind of a life history of chromosomal instability in these tumors. And what we see is these in Wilms tumor correlate quite nicely with different risk groups. So the high risk ones tend to have more. But one interesting phenomenon that we saw too is that when we actually just took only the high risk group and we split them into those that had a lot of alterations and those that had few alterations, we saw that the ones with a lot of alterations actually really relapsed early. So using this biomarker, we could potentially propose a way of taking this high risk group and splitting it into also a very high risk group potentially. So with that, I'd already like to come to my conclusion. I think I would say that it's clear that every cancer researcher understands that cancers are formed from an evolutionary process. But I think it's really important that cancer researchers have to have in the forefront of their mind that there's always an evolutionary explanation for what they're observing. And this is true of both the biology and it's also clearly true of the clinical implications too. So when a patient comes into the clinic and they've relapsed, that relapse has occurred through an evolutionary process. So it should really always be something that we're constantly thinking about and asking ourselves, how can evolution help us answer these important questions? What I'd also say is that there's many parallels with species evolution too. So if you're in the field of population genetics or evolutionary biology and you're interested in cancer, I think if you learn more and more about cancer, you'll soon realize that there's lots of ways in which you can find parallels between the two. So just an example from some of my own sort of broad research is, I mentioned I'm interested in whole genome duplication, for example. I remember reading that in plant evolution, whole genome duplication is quite important. And I think one of the theories of why it's important is that it's an example of response to stress. And actually, this is a really good analogy for how whole genome duplication can be important also in cancer, because cancers grow in very stressful conditions as well. So there maybe is a link there between evolutionary biology, for example, and cancer evolution. So with that, I just also like to wrap up by saying, of course, my lab's local. It's in Vienna, very open to discussion. This is my email, just my name at ccri.at. It doesn't matter if you're not from the cancer evolution field. If you think something's interesting, please get in touch and let's discuss. Maybe we don't understand everything that we each say, but I'm sure we'll find some common ground. And of course, I really have to do acknowledgement slides because, as I try to stress, I was involved in all these projects, but I certainly wasn't necessarily leading every single one of them. A big shout out to my previous postdoc supervisors, Andreas Atariva and Trevor Graham, who led most of the studies. The Wilms tumor studies were led by Will Mifsud and Kathy Pritchard-Jones, and of course, I had many, many other colleagues that were working alongside me, both in the dry lab and the wet lab. And I'd also like to, of course, acknowledge my current research institute, the St. Anna Children's Cancer Research Institute, my own team, and many other teams at the institute and institute as a whole, and of course, many different funding bodies that contributed to all these different studies. And with that, yeah, I'd like to finish. Thank you very much.